For quite a long time, and definitely in the last 4 years, we've heard a lot about biotechnology, viruses, stuff that happens in labs, stuff that doesn't happen in labs but we think does, and much, much more since the pandemic. Today, we're going to be talking about something that is legitimate work that will be done in the labs in a few years down the line in the future, and that is legitimately dangerous and scary, that other scientists are asking everyone to stop doing this work right away. Lots of scientists from across the globe are raising really loud alarms because we now have the technology to do something highly dangerous and completely strange, which is creating mirror life in the lab. All life on Earth is made up of DNA and RNA. More complex life, such as ours, has DNA, but there is no life that has no RNA. Bacteria, viruses, even viruses that are sort of considered to be the outliers for life or are a border case between living and non-living because they cannot replicate unless they are inside living organisms, they also do have RNA. And we've all seen at least images of DNA and RNA, how they are twisty and helical. What is most amazing is that all life on Earth is made up of DNA and RNA that twists in one direction only. These are images of DNA and RNA. DNA is double helical, it has two helixes, and we can imagine it as two ribbons twisting around each other. RNA, meanwhile, is much more simpler in structure and has only a single ribbon that twists. Technically, they're called strands, but we're going to be using highly simplified language through this video to visualize things. Now, there's an interesting thing to notice in this visual. As you can see, they twist only in one direction, the right-handed direction. When the DNA molecule or the RNA molecule forms inside of life or wherever, it builds itself up in such a way that it twists to the right. This is called chirality. Chirality is when something has right-handedness or left-handedness. Now, let me demonstrate. Now, I'm just using examples around me. Now, this is a cup. It's the print mug. And let's assume I am the mirror. You are looking at me like this. Now, here's a cup. And the reflection of the cup is going to look like this. These two are reflections of each other. The handle here is on one side and the handle here is on the other side. But is the object and the reflection identical? Yes, because this reflection can be twisted, turned across some axis and made to look identical to the original object. So these cups, this particular cup and its reflection are identical. They can be twisted and turned in some way to be made identical to each other, and therefore they are achiral. They do not have right-handedness or left-handedness. But let's do the same example with our right hands and left hands. Now, this is my right hand, this is my left hand. Clearly, if I'm the mirror, this is reflections of each other. They overlay as reflection. But there is no way I can twist and turn one of my hands to make them both identical. It just will not happen. We have a right-handed side and we have a left-handed side, and they have chirality. The right hand is not the same as the left hand. The object is not the same as its reflection, so they have chirality. This is the same for RNA and DNA. Now, these are our hands, and when something turns this way towards my right hand, this is my right hand, it is right-handed or is turning clockwise, and if it turns towards my left hand, it is anti-clockwise. So when something turns this way, it is clockwise, this way is anti-clockwise. This is right-handed and this is left-handed to me. Now, I'm just using objects around me to demonstrate. This is a piece of string light and this is a piece of discarded curtain rod. Now, I'm going to twist this rope light around this rod when it is right-handed. So, I'm turning in the right-handed direction and going this way. So, as you can see, To do this, you first have to turn the light to the right hand direction, whether looking at it from your end or my end. Now left-handed is going to be the opposite. I'm going to turn it the other way. As you can see, to achieve this configuration, the rope first has to be turned in the left side. This can also be easily visualized as banisters on a spiraling staircase. If the banister is to your right, if you're falling off to the right, then the stairs is going towards the left. And if you're falling off, if the outer edge is on the left, then you're basically going inwards to the right. And as you can see with these two light twists, at this point, with the two twists, we can see that the right-handed twist is different from the left-handed twist. And there is no way to flip or turn the orientation of one of these strands to make it turn the same way as the other, to make it identical to the other, which means they are not achiral. They cannot be twisted to become identical. They cannot be turned around any axis to become identical. They have chirality. So this is how all RNA and DNA have right-handedness. Similarly, these are what actually have right-handedness here is the nucleic acids, the bases that make up the RNA and DNA. Similarly, proteins and amino acids in all life have left-handedness. And this is just how nature is across the world today. Even when we encounter amino acids in meteorites, we find that the chirality is similar or is the same as what is found on Earth. Do we know why though? There are of course a few theories, and one of the most widely known one, widely accepted one is that there is selection bias for evolution. Large molecules with large right-handedness twisted themselves and just began replicating, and the entire biological system just aligned to work with it, and all the left-handed ones just died out. We see this even in the world around us. For example, screws. Any screw threads towards the right. That's why we say righty-tighty, lefty-loosey. If we turn screws towards the left to make them tight, that would just break the existing way in which we function. Across the world, screws screw on tighter when turned to the right. That's because they're threaded that way. That's because they work in the existing system, which is sort of the best analogy I can draw to life for this particular theory. Now, that is not to say that left-handed screws don't exist at all. Some very local esoteric systems do use it, and the same way, we do have DNA, sometimes within our own body, that turns to the left. So there are three types of DNA called A DNA, B DNA, and Z DNA. B is the most common and is found across all life. Both A and B twist right, whereas Z is an exception that is a little bit zigzag and twists towards the left. This DNA is usually found in people with diseases, as in the DNA occurs after people have diseases, especially neurodegenerative disease. It is considered an aberration. The rest of the body does not have that kind of DNA. But that's the beauty of it. The world survives on B DNA, which is the most common, and it twists right. In our world today, all bacteria, all viruses, all pathogens, and the molecules that target them, the medicines and their molecules, all of them are designed for right-handedness. At the end of the day, it is basically at a molecular level that this right-handedness evolves, and we can see this in the molecules that we use to treat our illnesses as well. When pharmaceutical companies and biomedical researchers engineer drugs to develop as therapeutics to treat diseases, they have to do so keeping chirality in mind because of the way these molecules latch on to the receptors in our body that are tailored to right-handedness. Ibuprofen is a famous example of a drug that has both left-handedness and right-handedness. One mirror image of the ibuprofen molecule treats pain and inflammation. It is commonly found, it's over the counter. I can just walk into a pharmacy and buy ibuprofen. But this ibuprofen works only if the molecule has one mirror image. If it's the other mirror image of the molecule, it does nothing to the body. It's not going to help me. That is a harmless example. But chirality is what we're talking about today because it can be dangerous. A more serious example is thalidomide. Thalidomide has been in the news for affecting pregnant women, and it is also used to treat morning sickness. So what is happening with it? One mirror image has the therapeutic value. The molecule, when oriented in one direction, helps morning sickness in pregnant women. The other mirror image of the same molecule leads to birth defects. Not so harmless after all. So the world exists on basis of this chirality. Now, in 2024, we have lots of amazing technology. In fact, we have technology that we can work on and improve for the next 10 years, and then suddenly find ourselves be able to bioengineer bacteria and viruses that can be left-handed in the labs. Basically, mirror image of what we have today, or mirror life. And this sounds very science fiction, but it's quite apocalyptic and it's quite real. In a new paper that came out this week, experts across the world penned an open letter outlining the large risks to life as we know it if we created mirror molecules of life. And they have asked to straight away ban all such studies, which are in their inception stages right now. They have asked for all such funding to cease and be stopped right away. Now, we might have an idea why, considering what thalidomide did. But these scientists do a much better job at outlining all of the risks to the earth as we know it if mirror life suddenly began to exist. This research has been going on for a while, and these group of scientists came together and consolidated all of the risks and explained it in this paper. This group contains scientists who are experts in synthetic biology, immunology, biosecurity, public health, microbial ecology, plant physiology, and much, much more. So what are they saying the risks are? If suddenly now there exists a left-handed bacteria in our world of right-handed life and its associated right-handed systems, what would happen? Well, the bacteria will be able to infect multiple species across large geographical areas because none of our immune systems would work against a left-handed mirror bacteria. Similarly, this is true not just for humans, but also for all other animals and all other plant life. Similarly, none of our antibiotics would work against this kind of specially engineered bacteria, as they all work on the mirror image. Microorganisms like bacteria and virus especially are not constrained and are not biocontained from their structure and surroundings just because they end up becoming left-handed. Even mirror microbes can find a large range of nutrition sources to feed out of and also infect and eventually kill. And they also persist for a long time. We have already seen this in synthetic biology, where reverse chirality of molecules leads to slower biological degradation and higher strength and resilience. So this life that scientists would create in a lab just for experimentation, for very valid reasons, to understand how chirality works, could potentially risk all life on Earth. The scientists who wrote this letter conclude that mirror life will be immune evasive and ecosystem invasive. Mirror bacteria and mirror life have the technical ability to break down every single kind of biodefense that the entire world has, because the molecules are different and will not fit into receptors for what we have. They can proliferate quickly and pose a large global scale biohazard. Within a year from creation of this life, we could have a worldwide pandemic that affects multiple species, including plants, and that we will have absolutely no treatment for. Then why would someone build something like this? What are the benefits? If the harms are so bad, why would scientists be experimenting with creation of a mirror bacterium? What are the benefits of mirror life? At this point, turns out, not really much. It is true that these forms of mirror life and working with them can help us create mirror therapeutics, molecules that we can study better and use them to treat conditions that we have not been able to treat before. So there is definitely potential for benefit, but these benefits and their potential are in no way even close to outweighing the risks. So everyone's worried. How worried do we have to be, you and I? It's not that we will have mirror life tomorrow or even by 2030. Creating mirror life in a lab is at least 10 years away. But what is important is that we are slowly making technical strides to that effect. But now, knowing that we can do this in 10 years, scientists are already asking people to stop and not take efforts to go in this direction. In the open letter, they explicitly say that all such work must be ceased immediately unless there is extremely compelling evidence that arises of their benefit. This is not expected to happen anytime soon. To stop all of this kinds of research, the scientists also outline different steps for the different involved stakeholders to follow. First, they say that all mirror genomes should be stopped, anything that can even create a cell that is a mirror cell. And then there should be regulatory measures that monitors all such progress in labs all across the world due to the unprecedented risks that they pose. And to this end, they say that it's not just scientists, the pressure also has to come from society at large to take responsibility to prevent the creation of this kind of mirror technology and mirror life that might pose risks at unprecedented scales.